Okay, so let's go back for just a second and look at this whole idea of reproductive isolation, because what we're going to be talking about today and in subsequent lectures is we're going to be describing how species form. Remember Darwin's theory and Darwin's natural selection by evolution. What we've examined about it so far was the idea of the population changing, right? And I know I must have said that 100 times, but it is one of the more common mistakes and misunderstandings that students have about evolution. It's not the individual that changes, of course. It's the population, specifically the genome of the population, the genetic makeup. So the reason why reproductive isolation is an important topic to discuss when we're talking about species is because if organisms continue to reproduce, all right, so if organisms in a population continue to reproduce with one another, then you're never going to get differences. Okay, now the population may change, but you're never going to get differences where you get two different populations. And eventually through time, what happens potentially is these two different populations become separate species. But that's all contingent upon the fact that they don't reproduce with each other. If they continue to reproduce with each other, then as I said, you're never going to get that. You're never going to get that division in the genetic information. You're never going to get that chance to have a species. So that's why we went through all these pre-zygotic and post-zygotic barriers to come up at the very end. And remember, Ernst Mayer's definition says that if at the very end of all this, they produce viable, fertile offspring, then those organisms are members of the same species. And if they don't, then they're members of different species. So all of this that you see here, the habitat isolation, temporal, the gametic, the hybrid fertility, it's all tied to reproduction. And it's all tied in terms of speciation to isolating reproduction so that you don't have that continual mixing. Now, let me show you what I mean. It might be a little bit helpful if I show you some examples of what I'm getting ready to talk about here. So we've seen all this. All right, so we're going to start off our topic today, our discussion today, talking about patterns of speciation. And what we mean by patterns of speciation is how is the genetic information initially divided? Right? It's kind of like what we were talking about in the founder effect. You remember the founder effect was the birds that just, by chance, by luck, made it over to the offshore island. Very few of them may have made it, and sometimes it's just one female with an egg. So that's a lot less of the genetic information that was in the original genome. But remember, I said that genetic drift was not considered adaptation because even though the birds that make it out to the island may survive and adapt and all that sort of stuff, it was how was the initial set of genetic information changed? How was that initial small population of genetic information, how did that come about? In the case of genetic drift, it comes about by luck, right? Or whether it's a boulder rolling downhill and just crushing over some of those unlucky flowers, or it was the bottleneck effect that we talked about, or it was the founder effect. We're talking about how initially the genetic information was changed. So what we're looking at here in patterns of speciation is kind of the same idea, except we're not talking about how the genomic information was changed. I mean, that is presumably what may happen. We're talking about how is this initiation of reproductive isolation started? Right? You got to have them reproductively isolated if you're going to create separate species. So the first thing we're going to talk about is how did that happen? All right. So there are two types of speciation patterns, and you can see what's happening in the picture here. We got a pond with a group of fish, all of the same species, all indicated by being, what is that, orange or yellow in color. And the two types that we're going to look at, the first one we'll look at is the one on the left, that's allopatric speciation, and then we'll look at sympatric speciation. But either one of those events is doing what I said. It's going to reproductively isolate the organisms. Let's look at allopatric speciation first, because that's actually one of the easier ones to understand, right? They're reproductively isolated because they ain't in the same place. All right, so at one time, this lake had these fish swimming in it, and there might have been this obstruction in the middle of the lake. And for whatever reason, the climate changed. An earthquake occurred that drained some of the water out of the pond, or whatever the case may be, the water level dropped. Okay, so that's what we're seeing down here with allopatric speciation. So now that the water level dropped, that part that was underwater and the fish could swim over at one time, now they can't, right? They can't make it over this barrier. So what the picture is trying to show you there is if this were to happen, right, this actual geographic barrier, what one of the possible outcomes, now one of the outcomes is the fact that the fish are just now two separate populations, although they stay the same species. What we would assume is that if the organisms were separated, in this case, let's talk about the ones on the left, because they're the ones that have changed into a new species. If adaptation is necessary, if there's some adaptation is necessary to adapt to this slightly different new environment or radically different new environment, then those organisms are going to change through time. First, they'll change and become, the population's going to change, and then eventually, like microevolution, those little tiny changes in the population are going to lead up to big changes that eventually results in a new species. Okay, so allopatric speciation, again, the way that the reproductive isolation was initiated in allopatric speciation is through a geographic barrier. All right, something formed or something occurred that physically separated these fish from one another. And as I said, and that doesn't automatically mean you're going to get speciation. So don't go in the backyard to your pond and build a wall between between all the bass that are swimming in the pond and expect the bass on one side to become something different. Because even though you've got a wall in your pond, the conditions are the same for the most part. And adaptation is contingent upon the environment. So the environment's the same, then, right? We've always said that if it ain't broke, don't fix it. If the environment stays the same across this barrier that you see in the picture, then there's no reason to believe that those organisms are going to speciate, become different species. Now, based on how they were broken up, this is kind of like the founder effect again. The population on one side might have a slightly different genome than the population on the right, or even more than slightly different, but it's not going to have enough of a genomic difference that we actually have a separate species, a different species. So we're going to talk about allopatric speciation for a couple slides, and then we'll turn our attention to sympatric speciation. Now, allo means other, and sim means side by side, okay? So in allopatric speciation or species are formed in an other place, the other side of the pond there. In sympatric speciation, as you can see in the picture, speciation occurs within what we say the ancestral population. The ancestral population in this case is the gold or the orange-colored fish, and you can see the blue ones are scattered in throughout there. So in sympatric speciation, it's a little different the way it works because you're not separating them physically. It's very easy to understand, reproductive isolation at the physical level. That was the habitat isolation that we were talking about in all those different reproductive isolation things that I was discussing the other day. Sympatric speciation, a little different, but we'll get to that in just a second. Let's turn our attention back to allopatric. So again, the allopatric speciation, the key is geographic. Let's look at some examples. Geologists tell us that the Isthmus of Panama, that little narrow chunk of land in Panama, arose or became prominent. The sea levels dropped is what basically happened here, folks, about 9 to 13 million years ago. Well, prior to the Isthmus of Panama being there was nothing blocking, these are shrimp, folks, of the genus Alpheus. There was nothing blocking these shrimp from swimming from, well, what became the Atlantic side to what became the Pacific side. There was no isthmus there, so they moved back and forth. There was no reproductive isolation. They reproduced with one another, so the genetic information was constantly mixed up. Now, initially there were, I believe, 15 species of, 15 species of these shrimp living in around this area, we believe, looking back through the fossil record. After the Isthmus of Panama formed, again, about 9 to 13 million years ago, we go back later on and we come up with these things that we call sister species. Now, sister species are organisms of different species, of course, but they are relatively closely related. And in the picture you see here, we're showing you two sets of sister species. There actually ended up being a total of 15 sets of sister species. So where we had 15 species at one time, after the Panama Canal, or after, not the canal, but after the Isthmus of Panama formed, we ended up with a total of 30 species. So these are sister species of one another. And then Alpheus nuttingi and Alpheus milliceae were sister species of one another. Alpheus formosus and Alpheus panamensis were sister species of one another. So what happened here is the formation of the Isthmus of Panama caused these species that were initially sympatric, in other words, they were living in the same place, to become allopatric. They're no longer in the same place anymore. And there's no reason to assume that shrimp could make the trek across the Isthmus of Panama to breed with shrimp on the other side. All right, so what we end up getting a situation here is these organisms that were initially, like I said, just 15 species, once the Isthmus of Panama forms, and then adaptation was necessary because there's a little different conditions, you know, on the Pacific side than the Atlantic side. So these species change to adapt to the uniqueness of their environments on either side of the isthmus, and we end up now having two species, right? And that happened basically over the entire 15 species. So now we have 30 species where we started off with 15. You see the reproductive isolation though, right? It's the island, or I'm sorry, not the island, but the isthmus that formed between them. Allopatric speciation. Now what's interesting, as we are learning more and more about genetics and being able to examine genetics, the best way, you remember when I was telling you about what makes a species and we went through morphology, you know, the way they look, ecology, but one of the ones I mentioned was phylogeny. And I said phylogeny is your family tree. It's who you descended from and who descended from you species-wise. Well, phylogeny, as of late, has been helped considerably by our advanced techniques in analyzing genetic information. So what these phylogeny folks have done is they've gone back and analyzed the DNA of these sister species of shrimp. And when DNA is modified and changed, many times it does so on a regular basis. So you can actually count up the number of changes. You know about how many years on average it takes to have one of these changes. So you can do the math and get a sort of a rough idea about when these species weren't separate species. All right, now, so I'm going back to this phylogenetic tree, you know, we got here, and then they diverged. You remember this is the same tree I drew for you guys the other day when we're looking at the horses and the dogs, right? We said the reason why horses and donkeys can occasionally make a fertile offspring when typically they make a sterile one is because horses and donkeys diverge from one another, you know, sometime around there. So just past the juncture between those two. If we were looking at the dogs, since dogs and wolves always make fertile offspring, even though they have, you know, the same species name, we would say that dogs and wolves are somewhere, you know, just a smidgen past where they broke. And remember, I told you that wolves were domesticated, ergo dogs, 10, 12,000 years ago. So that's like yesterday when it comes to evolution, right? So you have any questions on that, of what I mean, how this represents allopatric speciation? Now it's good evidence for allopatric speciation. At 15 species, throw in the isthmus, now we got 30 species. When we go back and we look at the phylogeny and we time when these species begin to separate from one another, right, what I was just talking about, we find out that these sister species, or these shrimp, begin to speciate, creating these other species, about 10 million years ago. Fits in very nicely with what the geologists tell us when the Isthmus of Panama was formed. See, that's what I love about being able to bring in all these different disciplines, and that's why science of any discipline cannot work in a vacuum, folks. Biologists need to interact with geologists, and geologists interact with chemists, and chemists with physicists, and all together that gives us a much better picture. because the geologist had done the research and been able to tell us about when the Isthmus of Panama formed, and because folks studying phylogeny, phylogeneticists, looked at the break genetically between these species, found out it occurred about 10 million years ago, we could say, that's a smoking gun. That pretty much indicates that it was the Isthmus of Panama that formed that geographic barrier that caused this allopatric speciation. I got a question for you, just as a side. If I was traveling from the Caribbean, I'm going to be drawing in the little map in the upper left. If I'm going from the Caribbean, my pen will cooperate, to the Pacific, when I come out of the Panama Canal, so I'm going this way, am I further west or east? So I'm moving from the Caribbean to the Pacific, or the Atlantic to the Pacific, and once I've gone through the canal, am I further west or east? Dustin says west. What do y'all think? Jared says east with a question mark. Why question mark, Jared? EW? East-west? Hedging your bets on both of them? Harrison, you said east. Anybody else? Harrison, why did you say east? Or Jared, why did you say east? Or Dustin, why did you say west? Well, 50-50 chance. Can't beat. You can see it in the picture. This, Pacific's on the west side. Right? Pacific's over here. Atlantic's over there. Here's the canal. Right, there's the canal. So what I'm doing is, as I go through the canal, I enter on the eastern side of the isthmus, but as I go through the canal, I actually end up not further west, but further east. The canal kind of goes back on itself. You see what I mean? So these are the north-south lines. So you're actually traveling from the east side of the isthmus to the west side, but on the whole global thing, you end up a little further east than west. My son likes to do those trivia things where you go and I get bars and clubs and they ask a bunch of trivia questions, and he comes home and he tells me sometimes, and one of them, the reason why I bring it up a couple of weeks ago, he said was this question, that the Panama Canal on the Pacific side is actually further east than the Panama Canal opening on the Atlantic side. Little trivial bits of information that probably will do nothing for you. Okay, Brittany, look up in the little map. In here, this is the east. This is the west side of the Panama Isthmus, that little thing in the middle, right? Yeah, okay. But now the canal doesn't run straight across. It actually runs back on itself. So what you do when you're sailing in, you actually, when you come out the other side of the Panama Canal, you're further east than you were west. See? Come on, pen. You see what I mean? You see how down here, okay, you see down here, hello, are you guys still hearing me? I feel like I'm locked. Okay. All right. Hello. Can you all hear me now? The heck was that? You. All right. Sorry, folks. For some reason, the entire program locked up, but I appreciate y'all sticking around. So back to it, Brittany. Again, so we're entering the Atlantic Ocean, which is on the east side of the isthmus, but you notice that you actually travel back. You're actually traveling back, right? So when you come out on this side over here, right there, you're actually further east. You got it? Okay. All right. Okay. Is recording? Yeah, recording's still going. All right. Here's other ways in which allopatric speciation can occur. So you can get this isthmus, get this landmass that forms. Well, one of the other ways is, look at on the left, we've got a stand of pine trees, all right? And they're all the same species. They're all the same color here. And a river forms between them, just like maybe the Grand Canyon. And that river that forms between them, the implication or the idea is that river is wide enough so that the pollen, right, the male part of the plant, the male gamete of the plant, if you will, the pollen from one set of pine trees, it's too far to make it over the canyon to the other pine trees. They can't make it as far, it's too far apart. Now, once again, if adaptation warrants it, if there's a little difference on one side than the other, then what we might get is a change in the population of the pine trees on the right-hand side of that river that I'm drawing there. And then eventually, through time, those little changes may add up to a completely different species. Now, this is still allopatric speciation because it still was initiated by geographic. It was still a geographic feature that initiated the isolating of reproduction. See, that's why I was saying the pollen couldn't make it over. You've got to reproductively isolate them, because if the pollen could actually, if it could drift over across, well, then you would get a mixing of the genetic information and you would presumably never get speciation. So it can be something like that isthmus to Panama, a river formed. Sometimes it's not that a geographic barrier arises or appears in an area where a population is found. Sometimes it's the population, as they disperse through the environment, as they move through the environment, they actually come to a geographic barrier that they have to go around because they can't go over it. So that's what I'm looking at here in the upper right. So we got population right there at the north side, if you will, of this geographic barrier. And as they disperse out, as they move out and the population gets larger, since they can't live on the mountain there, they move on to either side of the mountain. And then when they get back together on the other side, the populations are sympatric again, that is, they're in the same place. And if they interbreed and they produce viable, fertile offspring, then guess what? Speciation has not occurred. Right? So sometimes it's not a matter of the geographic barrier forming, it's a matter of the geographic barrier being a part of the actual distribution of the organisms. Now, in the other one, if the populations become sympatric again, in other words, they end up in the same place again, but they don't interbreed, then we can say speciation has occurred. Simple enough, huh? Neat little example down below involving the Grand Canyon, kind of like with those pine trees that I started off with. Let me talk a little bit about scientific names. These are chipmunks, and you can see their scientific names. Scientific names are Latin-based for the most part, so often if you're having troubles pronouncing the name of an organism, do it phonetically. You know, like you learn phonics. I guess, I don't know if they teach phonics in school still, but for example, the genus of these guys is Amospermophilus harrisi, one on the left, and Amospermophilus leucurus. So if you just kind of say all the letters, many times you'll be very close to pronouncing it the proper way. All right, well, they're both the same genus, but they're different species. So since they're the same genus, that means they're closely related to one another. We might use the same term for them as we use with the shrimp when we call them sister species. Now, if you've ever been to the Grand Canyon, the guides that take you around there will oftentimes discuss these chipmunks that are found on either rim. See, the harrisi is found on one rim of the Grand Canyon, and leucurus is found on the other rim of the Grand Canyon. Now, there's no way, as much as these chipmunks would like, that they can jump across the Grand Canyon and find a mate on the other side. So initially, those were two isolated populations. All right, now let me step back for a second, because I'm afraid there may be some confusion here. There may not be, but I just want to make sure. If we're talking about, say, for example, eastern gray squirrels. Eastern gray squirrels have the scientific name Urus carolinensis. Sometimes the scientific, I didn't capitalize Cirrus, I should have. But sometimes the scientific name, at least the second part, gives us some descriptive feature about these organisms, or perhaps it's oftentimes where they were first described from. If you discover a new organism, a new species, you get to name it. The genus that you give it has to fit with, all the characteristics it is to be in that genus. But the second word, you can say anything you want. My major professor in school worked on shiners, which are little tiny fish, and he did quite a bit of work on identifying different species of shiners. Many years later, somebody discovered a new species of a shiner, and because of all his work, they actually named it after him. His last name was Snelson. So it's called Notropis snelsoni. So years from now, if you discover a new species and you want to honor your Bio 2 professor, you'd call it whatever. We make it Latin, Clarkii. All right. Okay, so Cyrus carolinensis was described from the Carolina region. Oh, here's one for you. Here's a scientific name. See if you can tell me where you think this critter was first described from. Obviously, that's the scientific name for alligators, but where were alligators first described from? Yeah, isn't that cool? Alligator mississippiensis. Okay, so and now the eastern gray squirrels that live in Florida are the same species as the eastern gray squirrels that live in New York, right? And same ones in Central Park. Cyrus carolinensis, same species. They have the potential to breed, to make viable fertile offspring, but we would consider them separate populations. And the reason why we consider them separate populations, not separate species, is because of the chance of them reproducing with each other is pretty minimal, if not nil. So they are, for all intents and purposes, reproductively isolated. Now, I'm not saying that they're separate species. They're not. They've got the same name. I'm not saying they necessarily will become separate species. That kind of depends on the environment. That depends on adaptation. But if you've ever seen the squirrels in New York City, you know that there is a profound difference between the eastern gray squirrels running around Central Park up in New York and the ones we have running around campus. Have any of y'all ever been up there and seen the squirrels I'm talking about? Do you know what I'm referring to, the profound difference? It was one I remember. I went up there. I was like, holy cow, look at these squirrels. What was the... Oh, yeah. They look like small house cats. First time I saw it, I'm like, is that a squirrel or a house cat? Now, them being larger is because it's colder up north and larger organisms lose heat more slowly. So up north, that's a benefit. And down south, that would actually be a detriment. So that's why the squirrels are smaller down here. Now, that doesn't make them separate species, though. With time, maybe they'll become separate species. But at this particular point in time, they're just different. Well, sometimes they call them subpopulations. And this is where we sometimes get into this idea of what a subspecies is. We all know, for example, with human beings that our scientific name is that, right? Our full scientific name, though, is Homo sapiens sapiens. That's our genus, species, and subspecies. Now, a subspecies, two different subspecies are almost identical to one another because there is an actual other Homo sapien out there. Many anthropologists consider Homo sapiens. It's another Homo sapien, Homo sapiens neanderthalensis. The Neanderthals, who are no longer around. Now, we bred with the Neanderthals. We produced viable offspring with them. It was very common. They weren't a separate species, just a separate subspecies. Now, just for your own little knowledge, Neanderthals got their name from the first discovery of the skeleton of a Neanderthal was near a river in Germany, the Neanderthal River, I believe it was. But in German, TH is pronounced as a T and not a TH, not a th sound. So it's not a Neanderthal, it's a Neanderthal. Now, so what may happen with these squirrels, and for all I know, mammalogists have already done this, they may have subspecies them. They might be Sciurus carolinensis something or other down here, and Sciurus carolinensis something different up there. But a subspecies is not a separate species. It might become a separate species, but it's not one there. So these Amospermophilus, these chipmunks here, were at one time all living in one location. They were one population, one species, and then through thousands and millions of years, the Grand Canyon formed, and they become geographically isolated. And again, if adaptation warrants it, slightly different conditions on both sides, then you would get organisms to adapt and perhaps become a different species. Now, the idea is having different species names, if you artificially, if you stuck a chipmunk in your backpack and hiked over to the other side and let it go, that theoretically, that should not be able to produce viable fertile offspring with the Amospermophilus herocyte chipmunks. Okay? So allopatric speciation, the big thing is geographic isolation, a barrier that forms, a barrier that arises as organisms distribute themselves. But there's another way that allopatric speciation can occur. And oftentimes it happens in organisms like plants, these immobile organisms. Let me show you what I mean. Now, what we got here is one species of a typical conifer, a pine tree. And we're looking at 3 stands. That's what they call groups of trees. This picture's got 3 stands of trees. There's the one in the foreground, the one in the background, and then there's one about halfway through with the red X's that I'll talk about in just a second. Now, when these trees are all together in that area, the trees in the foreground, the pollen that they produced or the gametes, it's really too far away for them to make it to those trees in the background. So essentially, these 2 stands of trees here and here are reproductively isolated from one another. But they remain the same species. Well, the reason why they remain the same species is the fact that the same species of trees occurs in another stand, the one off here to the left. So we get genetic information being shared between here, between this foreground stand of trees, the middle ground stand of trees, and then they're sharing genetic information with the trees in the background. So again, even though the ones in the front may not be reproducing with the trees in the back, they are reproducing with the trees in the middle, who are reproducing with the trees in the back. The trees in the middle are also reproducing with the trees up front. So we get a mixing of the genetic information, which would deter speciation, right? It would prevent, presumably, speciation from occurring. What I'm trying to show you in the picture, though, is this is another type of allopatric speciation. What happens is if, I don't know, a landslide coming down this mountain here, or a flood, or maybe a disease that moved in and killed off all these trees. Now, since those trees are no longer there, the trees in the foreground and the trees in the background, they got no way to exchange their genetic information. So, and again, this would be warranted by adaptation. Remember, species don't change unless the environment dictates it. But if adaptation were to dictate it, or the environment were to dictate it, these could become 2 separate species of trees. And again, it isn't really a matter of a geographical barrier in between them. In this case, the geographical barrier really is just distance. And before, when those trees were in the middle, there was like a stepping stone between the 2. But now with those gone, the ones in the front and the ones in the back are pretty much on their own. Okay? Does that make sense? Cool. All right. Now we're still sticking with allopatric speciation, geographic isolation. One of the things that allopatric speciation can do in conjunction with this thing that we talked about earlier called the founder effect. And you remember the founder effect was a small subset of the genetic information. And the example I used was birds on a mainland flying out to an island that they normally wouldn't be able to make it. Remember, the founder effect is a type of drift. It's chance. So these birds that made it out to that offshore island did so by luck. There was a tailwind or storm or whatever we were talking about, and they happened to make it out to the island. And then because that island was an archipelago, I just love that word. I think that's such a neat word. I should name one of my kids archipelago. And because those islands were a group of little islands, once the birds made it out to one of those islands, it wasn't that far to fly to the other islands. And the beauty of the Galapagos, that archipelago, the islands are different somewhat. And the birds adapted to the islands and their differences. And the birds also adapted not to compete with one another. So we got speciation. Remember, the theory or the idea behind Darwin's finches is it was one species. Ernst Mayr said it could have been one single fertilized female and eventually became, depending on what book you look at, 13 to 15 to 17 different species of finches. So we started off with one species and ended up with a whole bunch. This is something we call adaptive radiation. The adaptive part is the reason why you get so many species is because adaptation is warranting it. It's a little different on each island, trying to reduce competition, so we get all these different species. And the same thing is being showed in this slide here. We're not looking at finches, though. We're looking at kingfishers. So what we might have on the island of New Guinea here is a mainland population of kingfishers, all right? And these kingfishers exist throughout New Guinea. We have kingfishers here in Florida. We have a belted kingfisher. Obviously different than the ones from New Guinea. There's no New Guinea kingfishers around. But those mainland population of kingfishers were the same species. They bred with one another. They weren't reproductively isolated, so on and so forth. Now, if you notice, there's a few islands, archipelagos, groups of islands offshore. So what we end up is birds flying over to these islands and becoming isolated. And you notice in the birds up at the top of the picture, we actually have two birds on two different islands, and the appearance suggests that they're slightly different from one another. And then there's another one in the bottom on another island, and it's a little different. Because of the fact that, and this is all founder effect that's going on, these birds making out to the island, but because in that archipelago there are slightly different adaptation pressures, what we do is we get organisms that are adapting to it. Now, most of the time, if organisms fly out of their normal habitat to a completely different habitat, they die, right? They die because there's nothing there to support them. But if they survive in situations like this, the founder effect of these few little birds making out to an island and then jumping from island to island can actually lead to what we've been talking about, this adaptive radiation. So we had one species of the mainland population of kingfishers that gave rise to those three, and I guess there's more than three, those three other species we see in the pictures of those kingfishers. And as I said, this often happens on the island chains, archipelagos, because you can jump from island to island to island in an archipelago. Once you make it out to that first one, then it's easier to move to the other ones. So people who study this kind of adaptation, if they see where we have this mainland population of an organism and then in the islands around it, there are all of these organisms that look like it but aren't the same species, we often suspect that that's what happened. It was adaptive radiation. You know, these critters kind of moving out to slightly different habitats. So we get a lot of diversity in organisms because of their moving out to these slightly different isolated habitats. And again, notice isolation is the key there. If these birds could fly back and forth on a regular basis, in other words, if it wasn't such an event to fly back and forth, then you would never get reproductive isolation. You never get another species. Okay? Any questions on that? All right, so that's allopatric speciation. Allopatric speciation is geographic. The population gets separated because of some type of a geographic barrier, be it mountain, river, or, as in the case of the trees, distance. Okay, now sympatric speciation. Sym means side by side. So as you can see what's happening in the picture there, we're getting a new species, the fish in the blue, in amongst the original population, right? So we don't have reproductive isolation occurring because there's some physical barrier in the way. we have reproductive isolation occurring for other reasons. And among animals, oftentimes that has to do with behavioral components. In plants, this can occur, this sympatric speciation can occur because plants can tolerate their genetics being monkeyed with a little more than animals can. Let me show you what I mean, because you guys talked about this in Bio 1. If you remember towards the end of Bio 1, your instructor talked about mutations to the DNA, specifically to the number of chromosomes. Like, for example, you learned about something called an aneuploidy. Any of you remember what an aneuploidy is? Come on, Bio 1 wasn't that long ago, folks. What's an aneuploidy? If I gave you a hint, someone with trisomy 21 has an aneuploidy. One less or one too many. There you go, Dustin. Yep, an aneuploidy is one, we say it's plus or minus one chromosome. Either you have an extra one, like in trisomy 21, or you're missing one like in Turner syndrome. Remember, Turner syndrome females only have one X chromosome. Females typically have two, but they only have one. All right, so aneuploidy. How about polyploidy? What does it mean when an organism, or what does it mean when something happens where an organism ends up as polyploidy? A polyploid organism has what in terms of the number of chromosomes? It looks like I put my glasses on. Many. How many? How many? Many. How many? Many. Whole nother set, folks. If you have three more or two more, it's just more of an aneuploidy, folks. But when doing a polyploidy, you know that typically it's this. There you go, Kelly, right? But what can happen sometimes is through a meiotic error, what we call non-disjunction. Remember, a non-disjunction occurs during anaphase in mitosis, or anaphase 1 or 2 in meiosis, in which the chromosomes don't separate. Let's go back and take a look at that and review for a second. So here's a condensed chromosome. Here's its copy. Here are spindle fibers. And during anaphase, these chromatids are going to be separated, right? In a non-disjunction, what happens, though, is you get spindle fibers only connected to one chromatid. If you remember right, during the cell cycle in mitosis, there actually is a checkpoint to make sure that all the spindle fibers are attached to the chromatids. Well, you know, just because you have a checkpoint doesn't mean that things don't slip through that checkpoint. So what happens in a non-disjunction is this spindle fiber doesn't pull one chromatid to that side, it pulls two of them. Now this is what would happen in trisomy 21. But if that happens with a whole nother set of chromosomes, you could end up with, say, a diploid sperm, or it could be a diploid egg, plus a normal gamete on the other end, and you get an organism that is 3N, triploid, right? Or sometimes in extreme conditions, both the sperm and the egg are diploid, and we get a tetraploid individual. Now, among animals, this is generally detrimental. This is generally death, okay? There are cases of human beings that are born triploid and tetraploid, and they're often quite challenged in their life physically, physiologically. You know, Mother Nature's got this delicate balance, and you know, having more chromosomes is not a good thing. You're supposed to have basically the right amount. Okay, now among plants, though, plants can engage in polyploidism. They can engage in this act of doubling the number of chromosomes, or luckily for us, agriculturally, we can stimulate them to do that. Now, don't ask me how they do that, but there's a way they can actually get plants to engage in non-disjunction of these chromosomes. But when this happens between members of the same species, intra-specifically, we call it autopolyploid. And if it happens between two separate but relatively related species, we call it interspecific, and that's known as allopolyploidy. This is the more typical one in plants, is allopolyploidy. So let's look at this example on the page, how that might occur. So we got species A and species B, each with a different chromosome number. Species A's got 6, and for species B has got 4. Now again, we're not talking about breeding a pine tree and a fern. Okay, we're talking about organisms and plants that are relatively closely related. They might be in the same genus, for example, or in the same family. They're relatively closely related to one another. Now, they both undergo typical meiosis, and they both end up with their haploid gametes. Now, typically what would happen is for the normal gamete on the left would be fertilized or fertilize a gamete from another organism that had 3 chromosomes, and the diploid number is restored, and everything works just like we learned in mitosis and meiosis. But what can happen sometimes in plants, especially those that are closely related, is gametes from 2 different species can actually fuse, and they form a hybrid. Now, does that mean that these organisms are the same species? Well, you tell me, because notice what it says about this hybrid. This hybrid is sterile. So if you were a biologist examining this and this result came about and they formed this sterile hybrid, what could you say about species A and B? Are they truly 2 separate species, or are they perhaps the same species? Why do you say that, Melanie? There's one word in there that gives it away, right? Or anybody else? Why? Right. Sterile. There you go, Kelly. Remember, by Ernst Mayr's definition, if you're the same species, you make a viable, fertile offspring. If you don't, you can make a viable mule that is sterile, typically sterile, right? So this hybrid is not going to be able to reproduce. But this hybrid can undergo mitosis, and it can even undergo meiosis. So when it undergoes mitosis or meiosis, we might get a mitotic error or a meiotic error, like non-disjunction. So in this particular case, what happens is the meiotic error, the mitotic error, actually doubles the chromosome number. So each one of these individual chromosomes, which you remember in mitosis and meiosis, that's the first thing that happens is you make chromatids, right? You copy the chromosomes. So in this case, the chromosomes are copied, but they don't go through the rest of mitosis, so they're not separated. So now when you double the number of chromosomes from 5 chromosomes, you now have 10 chromosomes, don't you? And now you've got an even number. See, the sterile hybrid can't undergo meiosis because you can't have gametes with 2.5 chromosomes. But the new species down below that is viable and fertile and is a hybrid now has 10 chromosomes. And it can undergo meiosis. All right, so we can get a completely new species out of that. Now, as I said, plants seem to tolerate this more. They seem to be able to deal with this more. And as I was telling you, we actually have, prompted this and actually encouraged this and actually got ways to stimulate this in plants. And it's really helped us agriculturally. Bread wheat, Triticum aestivum, this is bread wheat, stuff we make, stuff we make bread out of, no kidding, is actually something that we cultivated through time from a wild grass. This is the wild grass. Now, ancient folks were eating wild grass, but as time went on, we learned a way by using the tendency for these things to undergo polyploidism to create a much more nutritious and much greater yielding wild grass in the form of wheat. Okay? So allopolyploidy in this case, good, because we've been able to use it to stimulate and form to form this very important crop species, wheat, right? Okay, but in animals, it's usually a problem. So animals, when they engage in sympatric speciation, polyploidism does happen, but as I said, when you engage in polyploidism with animals, you usually very often are going to get a detrimental effect. You get a dead organism. So let's take a look at how sympatric speciation. So how can species arise in the same area? In other words, how can species arise in an area in which they're not reproductively isolated? So let's look at these organisms. These are Lake Victoria, which is in Africa. Lake Victoria cichlids. Any of you that are aquariums, you know that if you keep freshwater aquarium fish aren't always very colorful. And cichlids are an option for folks. Cichlids do have bright colors, so they give you a little more visual appeal in your aquarium, although cichlids tend to also be very aggressive towards one another. So what we're looking at here is two closely related species of Pundamilia. We've got Pundamilia, there's on the top, and we're looking at males in this particular picture, and we got Pundamilia nairi. So these Pundamilia fish living in this lake in Africa, these males, females select males on the way they look. And notice they do look different, right? The one on top is, is lighter with the bars, where the one down below has the orange tint to it. So females, just like many times female birds that we talked about, are selecting their males based on the way they look. So scientists had a very interesting time with this. The reason why they did is when they went and they were collecting these Lake Victoria cichlids, and I'm going to come back to this slide in a second, they found every once in a while a fish that looked like this, which looked like it was a mix. See, this fish looks like it's a mix of the Pundamilia, the blander colored, and the nairi, because it's got a little bit of the bland and the orange. And this is a hybrid. We wondered how that came about. If females are selecting fish based on appearance, then how did we get a hybrid? Well, they took these fish back into the aquarium. Such a neat study. They took these fish back into the aquarium. They put all species, males and females, in the aquarium together. And then they artificially lit the aquariums with orange light. So that was the only color light that was shining down. Well, under orange light, the males look alike, don't they? It's not real obvious which one has the orange color, which one does not. When they did that and they threw in females, females mated with males of either species and produced those hybrid offspring that I just showed you in that other picture. So in this case, the sympatric speciation wasn't a change in chromosomes. It was purely a behavioral thing. The females were selecting males based on the way they looked. And if they couldn't tell how the difference between them, they mated with either one. The hybrid that they found, the significance of it, is this is a Pundamilia hybrid that is found in turbid water. This is actually what we call the turbid water variety of the Pundamilia. What's turbid mean? Yeah, and rough water, how would it look if you're trying to look through it? Like if you had a mask and you dove down into the rough water, how would the clarity of rough water compare? It's very murky, right? The visibility is really bad. So the reason why these hybrids were showing up is the scientists were figuring out that in this particular case, the males and then the females that were breeding with them were in this turbid water where the visibility and the light wasn't as good. So they were making the same mistake, if you will, as when they brought them into the aquarium. So even though these are considered two separate species, because they have two separate species name, it's a very feeble mechanism that keeps them apart. It's nothing about the physiology, nothing about mechanical isolation, nothing about, even behavioral isolation when it's in turbid water. Behavioral isolation is only important in them when the females can make out who is who. All right, so this would be an example of sympatric speciation. Those hybrid forms that are arising are reflective of the fact that Pundamilia, and Pundamilia nairi not too long ago were the same species, and they've separated purely along the lines of a behavioral component. Neat experiment. You just think, man, that's so cool from an experimental point of view. Can you imagine the scientists? They said, let's try this out, and it worked. Wow, it'd be fantastic. We demonstrated what it is that it's keeping these fish breeding with members of their same species. because this is an observation they made. Like, look at this hybrid. Why, if we know they select based on where they would look, why would females make a mistake? Well, because they can't see them very well. Now, amphibians, who tend to tolerate polyploidism a little more amongst animals, can undergo a process by which they can actually form another species. This is Hyla chrysocoelus, Hyla versicolor. Hyla are the tree frogs. We have lots of hylids here in Florida. And if you're trying to identify that frog, if you want to know if it's a tree frogs have expanded digit pads. They have this rounded part at the end of their toes. You can see it really well in this Hyla versicolor. See that? That's how they're able to hang on to things. So that's a clue when you're trying to identify those frogs. If they got these expanded digit pads, then that may be a green tree frog. All right, so what happens in this particular situation is herpetologists, folks who study amphibians, have found that Hyla versicolor is really a result of meiotic errors that occurred in the male and the female of Hyla chrysocoelus. So instead of making haploid gametes, they made diploid gametes, and so they created a tetraploid individual. And whereas I said oftentimes that's going to be fatal, in this particular case it was not. So it created a different species. Now, Hyla versicolor is its own species, and they breed with other Hyla versicolor, and of course they undergo meiosis the same way, but the chromosome number is treated properly. So once you're in the Hyla versicolor, which each one has its own 2N, then meiosis creates N gametes like it's supposed to. But we can trace back through genetic evidence to suggest that versicolor came about through a double meiotic error through Hyla chrysocoelus. Again, amphibians tend to be able to do that and tolerate that a little more than other animals. Especially when we get up into mammals. I always hesitate in classes to say always and never. I'm not aware. Well, there is that one rat in South America that I think they said came about through polyploidism, but it's quite rare among mammals. Mammals, if an offspring is conceived to be triploid or tetraploid, usually it dies, it doesn't survive. All right, so allopatric speciation, geographic is the isolation mechanism. Sympatric speciation, something other than geographic. It could be polyploidism. It could be behavioral, like it was in the cichlids, right? So that's what separates those two. Okay, now let's look at hybrids for a second. If you think about it, all of these organisms breeding out there, including those organisms who might breed with closely related species, you would think that eventually there wouldn't be hybrids, or I guess another way of saying that everything would be a hybrid, and that's not the case. Hybrid zones are a very interesting thing for population biologists and zoologists to study, because they're basically living laboratories. They're showing us speciation in action. Let me show you what I mean by that. So on the left, we start off with a population, 3 populations, all the same species, and they're all sharing genetic information. So genes are flowing between them. All right, so we've got the gametes of one population mixing with the gametes of another. So even though these 3 populations might be separate from one another, they're like those trees in the picture until those trees in the middle died off. All that information is being shared. So these 3 populations collectively would represent one genome. Now, each population has its own genome, but since they're cycling this genetic information and sharing it, it's all collectively one genome. And some barrier to gene flow occurs, a physical barrier, allopatric or sympatric, some sort of barrier. And one of those populations becomes isolated and diverges, right? And it becomes a different species. Again, warranted by adaptation. Remember, I've said a million times, species aren't going to change if they don't need to, right? Species aren't going to evolve unless adaptation is necessary. And adaptation is necessary only if the environment dictates it. But the other 2 populations, they continue to be the species they were. And if these isolated populations and these original populations live close enough to one another, then very often, depending how far down the path they are, they can oftentimes create hybrids. And we call this in a hybrid zone. So when two closely related species meet in this hybrid zone, three things can happen. Number one, the species differences can be reinforced. All right, so species A up here at the top stays species A, species B remains species B. Hybrids form, but oftentimes they're not viable, so there's not very many of them. And eventually through time, if reinforcement continues, there's going to be no hybrids. See the part in the purple, how it's getting narrower as we move to the right? That's indicating that hybrids are going to die off. So that's what I mean by a living laboratory. We can see how species arose by looking at this. It's so neat. You can say, okay, species A went this direction, species B went this direction. We look in the fossil record, there were hybrids for a while, and then they begin to thin out. All right, so that's reinforced. The differences in the two species was reinforced. Let me show you an example, then I'll come back to fusion and stability in a second of reinforcement. These are birds in Europe, flycatchers, closely related species. There's the pied flycatcher, the collared flycatcher, and these are all what the males look like. Females do the selecting, so there's mate selection in these species of birds. The pied flycatchers are found in the northern part of Europe. Collared flycatchers are found typically in the southern part of Europe, okay? It's in the yellow part where the hybrids are formed. Now, this is an example of reinforcement. This is an example of where you're not reinforcing the hybrid, you're actually selecting against the hybrids. Hybrids are very rare in this yellow area. For one thing, we think they have less fitness than parents. But they're also very rare because birds in the middle, all right, birds that live in that yellow zone there, the females especially, when females are choosing mates in this sympatric zone meaning they're together. In other words, there's pied flycatchers, there's collared flycatchers, there's pied flycatchers that are a little browner in color, and then the collared flycatchers, you see, they're a little different than the collared flycatchers in the hybrid zone than the ones that are in the blue. See the white around the neck? So in those hybrid zone, the females are less likely to make a mistake and mate with somebody. In other words, a female pied flycatcher is less likely to mate with a collared flycatcher and vice versa. And evolution has made it easy for these females because in the hybrid zone, if you're a female living in that yellow zone, pied flycatchers look a lot more different than collared flycatchers than the other original type of pied flycatchers look. You see, pied flycatchers up here are black with a little white. Collared flycatchers are black and the white extends back a little bit, but in the hybrid zone, the pied flycatchers are brown in color, so the females avoid them. The same is for the collared flycatchers. So since females recognize those organisms in the sympatric as not being members of their own species necessarily, then they're less likely to breed with them. And as a matter of fact, most of the time, a pied flycatcher female who's in the middle zone, if it sees one of these versus one of these, is going to pick this guy. And a collared flycatcher female in the hybrid zone is generally going to avoid this guy and pick this guy. So what we get is a lack of hybrids, right? Because hybrids would mean that the females would mate with pied flycatchers, the brown colored pied flycatcher, the collared flycatcher with a little bit more white around it, and we get this hybridization. And that would continue to encourage hybrids. But we call this reinforcement because since the females are doing the selecting, they're selecting against mating with those pied flycatchers and collared flycatchers that are in the middle, the ones that are brown, and the one in the collar that has the collar that goes all the way around. This is natural selection, folks. This is evolution encouraging the lack of hybridization because the males in the hybrid zones look different than the males the females would find if they were in the allopatric zones. A female up here would only see pied flycatchers that look like that, and a female down here would only see pied or collared flycatchers that look like this. It's in the hybrid zone where the confusion can occur. So evolution has created or evolved organisms that look not only or they look different in color, even though they may look different in terms of the way the body pattern is. All right, so that's what we mean by reinforcement. And then through time, like I said, hybrids will eventually die off. Now, on the other hand, hybrids can occur, and we get two different species, and hybrids occur between them. And then, for whatever reason, the two species that diverged come back together again, either allopatrically, the river fills in, and no longer is there the gap, and the chipmunks can make it across. But when fusion occurs, we lose the hybrids, and we also lose the separate species. Let me show you. This is a good test laboratory as well, or test in a laboratory. Let's look at this species, Insantina schultzii, the North American salamander. It's what we call a ring species. All right, so we're starting up here at the top with an organ population of this salamander. And as those organ populations of salamanders begin to move south, they came across this mountain range, the Sierra Nevada mountains in California. And a mountain range is a relatively formidable geographic barrier to these salamanders. So they went not through the mountain range, but for the most part, they went around it. And they ended up diversifying into a coastal population, the yellow-eyed and the Monterey, and then the inland population, the Sierra Nevada, the yellow-blotched, and the large-blotched. Now, notice what I said there. I called them populations. I didn't say the yellow-eyed or Monterey or Sierra Nevada were different species. They're different populations. It's still the same species. From a hybrid, somebody studying hybrid about this, the interesting part would be as they continue to spread out, because you notice, part way down, you can see that the coastal and the island populations occasionally would interbreed if they could, be lucky enough to find each other across the mountain range, and they produce fertile hybrids. So they're still the same species. But as we move further south, and of course moving further south is also moving further in time, because it took a while for these salamanders to make it down there. It could be thousands of years, for example. Somebody studying this would be interested, what happens when they get around the southern end of the mountains and the populations come back together again? So in those areas around the southern end, in which we have these coincident ranges, which the large blotched and the yellow blotched and the Sierra Nevada and the coastal and the island populations mix again, what happens? All right, so in areas of coincident ranges, no hybridization occurs. So what we're looking at in that species is what we call fusion. See, since no hybridization occurs, these organisms, as they made their way down either side of the Sierra Nevada mountains, and met again on the southern end, when they bred, they produce fertile offspring, fertile, viable offspring, not hybrids. All right, so we get fertile hybrids in between, and then down at the very end, the reason why they're not hybrids is because everybody's all mixed together. So if you were studying to see, whoa, have these salamanders actually speciated? No, because once they get to the southern end and they come back together again geographically, they're able to breed and produce viable fertile offspring. So that means that they never truly became a separate species. They stayed as different populations. Somebody naming these might call these different subspecies once again, like I was showing you earlier, but they're not different species. Okay, now the other thing that can happen, go back a couple slides, is we get stability. And in stability, what we basically have is the two species and we maintain the hybrids. For example, the wolfen that I showed you, the Tursiops and the Sidorca, the false killer whale and the dolphin. You got the line that is the false killer whales, you got the line that is the dolphins, but there are hybrids in between, and they're not dying off, and of course the two species come together, they're not the same species, so we're stabilizing in terms of a hybrid zone with those organisms. Dolphins, false killer whales, and then the wolfens in between. Questions on that? Oh, good timing, perfect. Right. Now we're going to talk about something Darwin didn't know. You remember I said at the beginning of this chapter, I think it was, if T.H. Huxley wrote the quote, and he said if the gaps between the fossil record could show to have always been there, that's going to be fatal to Darwin's theory. If the differences between species were always there, then we're back to day one in class talking about Aristotle and the rungs of the ladder. But the problem, or the thing that caused Darwin so much consternation, was that thing I was explaining to you the other day, the Cambrian explosion. 520 million years ago, when we look in the fossil record, we went from before the Cambrian explosion, a few species, to afterwards, it was certainly among the animals and among other creatures as well, to almost all of the major groups of animals we have on Earth today. Now, I didn't say all the species we have on Earth today, the major groups, the different kinds of worms, the insects, and all that sort of stuff, the arthropods, all of that arose in about 20 million years. Now, Darwin had spent his life, remember, understanding geology so that he could say, well, evolution works very slowly, and it needs millions or billions of years to occur. So the 6,000-year-old Earth could not be. And the geologists were the ones that helped him out with that. The processes that shape the Earth, gradualism and uniformitism and all that stuff we talked about are slow, steady processes. And given billions of years, you can carve a canyon or you can build a mountain range. In terms of biology, in terms of evolution, the type of evolutionary pace, as with the title of this slide, or pattern that Darwin was describing or was aware of was something called gradualism. And you can see gradualism on the left. And the way gradualism works basically is you got one species down here, and through time, those species become, populations of those species become isolated. And if adaptation warrants it, right, species don't change unless they need to, then as we make our way to the present time, we would have two separate species. Now, you could trace this back very easily in the fossil record. If you had enough fossils, you would have fossils going back in time that would be more and more similar until eventually you found a place in the fossil record in which you only found the fossil of that one bird. Now, gradualism does occur. Nobody's saying that Darwin was wrong. But the reason why Darwin had so much trouble with the Cambrian explosion is he didn't realize that there's another variant to the pace of evolution called punctuated equilibrium. All right, now punctuated equilibrium says we start off with this species down here, and for many, many millions or thousands or hundreds of thousands of years, that species undergoes little or no change. If it undergoes any change, it's going to be population change. It's not going to become another species. So that's the equilibrium part of the name there. But then in a relatively short period of time, and relatively is geologically, within 1 million years or so, some environmental condition occurs that causes the original species to branch off and form another species relatively rapidly. Now, the line being drawn horizontal across, if we're looking at time, is a little bit misleading. We ought to, it's not going to happen in one day. This orange-colored bird didn't one day jump up and become a yellow-colored bird. So really, this ought to be drawn more with a little bit of an angle. So with a little short period of time, we get that species of bird. And then maybe that yellow bird changed a little bit or not at all, maybe gradually. But in the case of the original orange bird, then a little bit further down the road, we get another speciation event that gives rise to these birds that have the red color. So in the end, we end up with 3 species. The original one, the red and the yellow. Punctuated equilibrium, folks. Species change little or not for long periods of time. Punctuated or dispersed in amongst that are periods of rapid speciation. So it's not just gradualism like Darwin was talking about. It's punctuated equilibrium in many cases. It's not just gradualism. That's exactly right, Melanie. Yep, it's like a family tree. So you might travel along this family tree for long periods of time, and then all of a sudden, in a short period of time, you get a bunch of species coming off of there. Punctuated equilibrium. Now, punctuated equilibrium is, again, both of these occur in evolution, but punctuated equilibrium does wonders for answering one of the biggest questions that evolutionary biologists have had to deal with. This is my last slide. Now, I may run a minute or two late. I'm sorry, but let me get through this with you. Later on in the semester, we'll talk about fish and amphibians, reptiles, and we'll also talk about how the evolution of amphibians occurred from fish, and how the evolution of reptiles from amphibians, and then how mammals and birds evolved from reptiles, okay, and how that all occurred. And we know that, you know, in the certainty of a scientific theory, that's the way it happened. But a lot of time the argument for that was, where are these half fish, half amphibian critters, if this is the way it happened? Where's the, you know, part mammal, part reptile? Where are these what we call transitional fossils? Where are these transitional organisms? So if we take for fact that birds evolved from dinosaurs, and I don't know if birds evolved from this dinosaur. I don't think it was that dinosaur. I just put dinosaurs up to be dinosaurs, and dinosaurs were reptiles. So if birds evolved from reptiles, that's kind of really wrong now, because birds are reptiles. But if this very specialized reptile, this bird, evolved from your typical reptile, then there should be an animal or animals somewhere in between that are part reptile and part bird. And for many years in the fossil record, we didn't find them. And people thought, well, if they don't exist, then this theory falls apart. And the reason we didn't find them is because if you go back to that previous chart, these transitional forms that might exist along this branch, they weren't around very long. So when you're looking at, for fossils, you might find, you know, 10 million fossils of this of dinosaur, 10 million fossils of birds, and you might only find a couple of 100,000 fossils of the Archaeopteryx. Archaeopteryx is a transitional form. Archaeopteryx, and there's a fossil of it, and we only have a handful of these. Archaeopteryx was part bird, part reptile. It had claws, it had teeth, and it had a tail like a reptile. But it obviously had feathers. It didn't have flight feathers. The feathers were actually used for warmth. The Earth was getting cooler during this time. That's why one of the reasons why the reptiles, the dinosaurs, were dying off. They don't do well in cold temperatures. But the warm-blooded critters, like the mammals that had fur to keep them warm, and the birds with their feathers, allowed them to survive the warm conditions. So punctuated equilibrium helps us understand why it was hard to find these transitional fossil forms, because there were very few of them. Archaeopteryx was around only a few million years or so. I don't remember exactly the time frame. Whereas the organisms that came before and the organisms came afterwards had been around a lot longer. Remember, finding fossils is relatively rare. So it's nice we can put those two theories together and use it to explain this whole question, this whole problem we had as evolutionary biologists. Where are these half critters? And oh, by the way, recent evidence, a lot of fossils being dug up recently from China have shown there's a lot more intermediate bird reptile critters out there. We'll talk more about those when we actually start discussing the birds later on in the semester, okay? All right. That's all I got for that chapter. That's, what chapter are we on? Chapter 24. We'll do chapter 25 next week. We're going to do lab 2. So make sure you bring that lab to class.